So today we are going to start our chapter number 9, that are the last steps of central dogma. In our last lecture we discussed about replication, that was the first step of central dogma, and now we will discuss about second and third steps, that are transcription and translation. Actually, transcription and translation combined together are termed as gene expression. So gene expression is kind of a process in which you can say protein and RNA and DNA work together to express the information that is in DNA. You know that the information that is stored in the DNA is called as genotype, right? And when that information is expressed, then the expression is called as phenotype. For example, if you have green eyes, or if you have brown eyes, for example, brown eyes are more common, and if you have brown eyes, then you have the genotype of brown eyes in your DNA. When your kids also have brown eyes, then that means your genotype was also expressed, your DNA was also expressed in your progeny or in your kids. So that was expression of your genes, right? So the expression of gene is called as phenotype. So the whole process is connected by proteins, right? So you can say proteins are the link between genotype and phenotype. So protein are the things that connects genotype and phenotype, and gene expression or protein synthesis actually includes transcription and translation. So we will discuss about transcription and translation in detail in this chapter. So these are the topic by topic yield in front of you, and you can say that transcription and translation are the most important one, while their next steps about RNA processes, types of RNA, they are less likely to be asked, but still every single topic of this chapter are important. Like, you know, you can see that there is no low yield topic in this chapter, right? So you can say that the whole chapter and every single point is important. Actually, the whole central dogma chapter is really important. There could be a question asked from any topic. You can skip some topics like low yield topics. I have told you about, like, in the last few lectures, like this topic would be neglected, this chapter or this topic can be left behind for IMAT, but there are so many topics in these chapters and none of them can be neglected. So you have to be really careful when you are solving MCQs from these chapters, because they are mostly conceptual. Yeah, obviously there are going to be memory based MCQs about what kind of enzymes are used, what is sense strand, what is antisense strand, what is a template strand, what kind of RNA polymerase 2 function, and this kind of, obviously this kind of MCQs would be asked, but most of it would be conceptual. You have to be really careful about solving these MCQs, and they have really good conceptual clarity. So the first topic is going to be about transcription. So transcription is actually the synthesis of RNA from DNA. You can say that the conversion of DNA into RNA is called as transcription. So this is the step that you have to keep in mind. Next thing is that it is the first step of gene expression. It is not the first step of central dogma, but first step of gene expression, and it actually occurs during three phases. The first phase is G0 phase, the next phase is G1 phase, and G2 phase. These are the three phases of cell cycle during which transcription actually happens. So G0 phase, G1 phase, and G2 phase, these are the three phases of cell cycle during which transcription will actually happen, and it requires actually only one single enzyme, and that enzyme is called as RNA polymerase enzyme. So RNA polymerase enzyme is the enzyme that will be used in the whole process. However, it is a continuous process. Just like replication we discussed in our last lecture, transcription is also a continuous process. Transcription and replication actually have same name of their phases. Just like replication, the first phase was initiation. Similarly, as you can see here, transcription also has the first phase, that is initiation, and this was the diagram that we used in your replication reaction, right? If you remember. So that's, you can say that the few steps of transcription and translation are actually really similar but there are many differences. We will discuss about those differences in the end of this chapter in a table, but you have to see that there is a bubble here, there is also initiation, there is also elongation, there is also termination, just exactly like the replication process. So first of all, that was the overview of this lecture. So you can say these are the phases, these are the enzymes. Now let's start our chapter and study about the whole process. The first thing is that you have to know what are the strands and what are the coding strands. So the template strand is the strand on which the whole reaction will actually happen. As you can see here, there are two strands, that this is a dark one and this is a light one. So the dark one, this strand is called as the template strand. This is the strand that would be copied by the RNA molecules. So first of all, this is what you can say it is called as template strand. It is also called as antisense or non-coding strand. Why is it called as antisense or non-coding strand? We will discuss it in later. The other strand is called as coding strand. As you can see here, this is the light strand that is in front of you. This light strand is called as coding strand, or you can say as non-template strand. So now you have template strand and non-template strand. So what is the difference here in both of these? Why one is called antisense, the other is called sense strand? Well, the reason is simple. For example, if you have a template strand and the template strand have adenine, thymine and guanine, what will be the RNA molecule? So RNA molecule will be adenine will be have. What will you have with adenine? First of all, let me ask this question. What will be with the adenine? You will be thinking about like, yeah, adenine will have thymine, right? But no, you are making an RNA molecule, so you will have a uracil molecule. You have to be really careful about it here. Okay, you know that there is no thymine in RNA molecules. So if you are making an RNA, then you have to keep in mind that you will have uracil, and with thymine you will have obviously adenine. So you will have reverse copies here, and with guanine you will have cytosine. So this is the going to be RNA molecule, and this is the what is it is the template strand that on which the whole transcription process is happening. So this is the template strand. So as you can see here, the RNA molecule that is being made on the template strand is actually the opposite of template strand. And if you make the non-template strand on which the process is not happening, if we take this strand, right, if we take this strand here, it will look like this. Before opening this molecule, what was in front of you, in front of adenine? Obviously it was thymine, right? And in front of thymine it was adenine, and in front of guanine it was cytosine. So this is actually as non-template strand. If I'm trying to make this topic as easy as possible for you. So now you have template strand and you have non-template strand. Obviously template and non-template strands are the actual part of the DNA molecule, right? So obviously the DNA two strands will have opposite nucleotide. Adenine will always be with thymine, like here, and thymine would be with adenine and guanine would be with cytosine. But you opened up the DNA molecule and you started making RNA in front of the template strand, right? It wasn't called template strand when it was a DNA molecule. It was, we started calling it the template strand during the transcription process. Okay, like DNA obviously has two strands. We don't call it a template strand, non-template strand. We only call it template and non-template when we are doing transcription or replication. So we call one a template strand and the one that is not being involved as non-template strand. So let's come back to the work we were talking about. Remove this. Okay, so as you can see here, non-template strand is just like the new RNA molecule. As you can see here, adenine, cytosine, and obviously thymine and uracil are actually the same parallel molecules. They are attached with adenine. So the template strand is actually the one that is being involved, but the RNA molecule that is being formed is just like the non-template strand, but in the form of uracil instead of thymine. I think we have made a lot of basic concepts here already. We have cleared so many MCQs confusions already at the start of this chapter, because so many students get these MCQs wrong in their final IMAT exam. So that's why we will say that the template strand is called as the antisense strand. Let's come back to the topic here. So as you can see, the template strand is called as antisense and non-coding strand. Why is it called antisense? Well, because the RNA molecule that is being made is actually like the non-template strand. That's why non-template strand or the coding strand is called as sense strand. So that was the reason why you say that non-template is called as sense strand or coding strand, because so many students get this wrong. Like they think like, okay, so RNA is being copied from template, then if we are making sorry, not being copied but is being made in front of the template strand, then the template strand should be the coding strand, it should be the sense strand, but it is actually the opposite, right? So RNA molecule is being formed on the template strand, but it has parallel nucleotides to the non-template strand. I think I made it really clear. If you still can't get the molecules or get the concept here, please go back in the lecture, listen to it again, and I am definitely sure you would understand it, because this is as much simple as we can make this concept. Okay, so that's why you can say that the template strand is called as antisense strand or non-coding strand, while coding strand or non-template strand is called as sense strand, because RNA is just like the non-template strand. So this was about the basic idea about what you will discuss here. So let's talk about the RNA polymerase enzymes that are being involved here. So RNA polymerase is the enzyme that is responsible for the whole process. Like unlike DNA polymerase, RNA does not require a primer. You know that in replication process, DNA polymerase actually required a primer, but RNA polymerase does not require any primer, and it can like do the synthesis on its own. And in eukaryotes, there are 3 types of RNA molecule: RNA polymerase 1, 2, and 3. The functions are in front of you. The RNA polymerase actually synthesizes ribosomal RNA. RNA polymerase 2 actually synthesizes messenger RNA and most small nuclear RNAs. And RNA polymerase 3 actually synthesizes transfer RNA and 5-SR RNA. And in prokaryotes, there is a single RNA polymerase with a sigma factor for promoter region, and it carries out all the transcription. Okay, so in prokaryotes and eukaryotes, that is the whole difference here. If we talk about RNA polymerase, so you have to be really careful about what to say it, how to say it. Like before we move on to next topic, let me make a diagram for you. So RNA polymerase actually contains 4 subunits. The first is 2 alpha and 2 beta. Let me draw it for you. This is first beta molecule, and then you have 2 alpha molecules like this, 1 and 2, alpha and alpha, and then a beta molecule again like this, and at the end you have another let's choose blue color, you have a sigma molecule like this. So actually the RNA polymerase contains 4 subunits, 2 alpha and 2 beta molecules, and they are called as core enzymes, while there is a sigma factor that is required to for the RNA polymerase to actually start the process, right? So the core enzyme and sigma factor combine together and they form a holoenzyme. You have studied about holoenzyme and apoenzyme, so you can say that subunits plus adding sigma factor, you will have a holoenzyme. What if we remove the sigma factor? You will have, obviously you will have an apoenzyme. So that was the thing that you have to keep in mind. So first thing is that once the RNA polymerase is attached with the chain and have started working, the sigma factor is actually removed, and the whole core subunits, the 2 alpha and 2 beta, these factors will start the reaction and catalyze the whole process. It is actually similar to the DNA polymerase in that it is also adds nucleotides to the 3' end. You can see here it is actually starts from 3' end. So as we have studied that DNA polymerase actually adds a molecule starts from 3' end, also same is for RNA polymerase. RNA polymerase also starts from the 3' end, but the difference between we have just discussed is that DNA polymerase actually require the primer, but RNA polymerase does not require any primer to perform the activities, right? In prokaryotes, only one type of RNA polymerase is found, while eukaryotes there are 3 types of RNA polymerase. We have just discussed them. So let's start our whole process that we had to know about. So first step is the initiation phase. So first of all, the initiation phase starts when transcription begins with the binding of RNA polymerase at the promoter region. You know that there is a promoter region where the whole step will start, and you will get a regulatory region of the gene that will combine or compress the binding site to the attachment of RNA polymerase. You will compress the whole site and RNA polymerase will actually attach. There are actually different kind of sites, or you can say binding sites, in prokaryotes and eukaryotes. We will discuss about eukaryotic binding sites. We will not discuss about eukaryotic binding sites because they are not really important. So the eukaryotic binding sites are actually TATA. They call it TATA box. This is the first one, and it is also called as 25 sequence. Then there is another sequence that is called as CAT box. This is CAT, CAAT. So it is thiamine, adenine, thiamine, adenine, cytosine, adenine, adenine, and thiamine. So it is called as 70 sequence. So these are the sequence, and these are the name of the sequence, and they are referred to the position in the promoter region that these sequences are located approximately how many nucleotides before the transcription starts. Okay, so these are the promoter regions, and these are the regions where the RNA polymerase enzyme will actually start working. So we have already discussed about RNA polymerase, what type of processes that it has, how it binds. So it will binds to the promoter and it will start to unwind the DNA strand. The next process is the elongation phase. So what happened in initiation phase? You had a promoter region in eukaryotes that was TATA box and CAT box, and on that region RNA polymerase came and it binded. And the next step is the elongation phase. As RNA polymerase actually binds to the promoter, it begins to arrange and polymerize the whole complementary template strand of the DNA, and it does not require primer to initiate polymerization first of all. And one of the 2 strands of the gene will act as a template, right? We have already discussed it for the transcription, and the template strand what we will call it? We will call it antisense strand, because messenger RNA would be complementary to that strand, right? You have to be really careful with wording in these chapters, so don't get any single MCQ wrong, because wordings can change a lot of things here, whether it's DNA, whether it's RNA, whether it's template strand or non-template strand. So in elongation phase, RNA molecule will actually keep on moving from 5' end to the 3' end of the RNA molecule, right? So we will move from 5' to 3' end of RNA molecule. Okay, you have to keep in mind, nor the template strand. For template strand it is 3 to 5', but for RNA it is 5 to 3' end. Both are opposites, right? I'm saying you there are so many things that you have to keep in mind. So RNA polymerase will keeps on moving from 5' to 3' end of RNA towards the terminator region of the genes. So we have already discussed about the terminator region, where the gene terminates, where the gene ends is called as terminator region. So besides its transcription bubble also moves along the DNA. So just like a replication, the transcription bubble is also formed, that it will also keeps moving towards the next position. and this event will continue until the RNA polymerase is actually what happens, reaches the terminator region of the gene. You can say this is the terminator region, this is the terminator region, and this is also the terminating region. So that's what happens in the elongation phase. Right. So what actually happens in the termination phase? So in the termination phase, the sequence of terminator region of the genes actually stops the synthesis of the RNA molecule. You can also say as messenger RNA molecule, and the terminator region will consist of a series of guanine and cytosine series. That is really important to keep in mind. And this pair will followed by a series of adenine and thymine base pair. So first of all, the terminator region will consist of what? Guanine and cytosine series and adenine and thymine series. So you don't have to memorize them. I am just telling you that these are the base pairs that are found in terminator region. So only for your knowledge. So first of all, that was the terminator region. So the part of messenger RNA which is transcribed in this region will project to form a loop-like structure. You can say that loop-like structure would be called as GC hairpin. Like it will form a structure like this. Let me draw it for you. This is the transcription bubble that is going there, and then at the end you will have a structure like this. So this is the structure here, and imagine this is a template strand and this is the RNA molecule that is being formed. So RNA would form a structure like pin-like structure like this. Okay, so if we draw it in a bigger diagram, it will look like this. So I have moved this diagram here so we can complete the diagram. So as you can see here, the RNA molecule will form a pin-like structure that we will call this as GC hairpin. We will call that is GC hairpin. So GC hairpin will be followed by a small tail of U nucleotide, uracil nucleotides. So it will have a tail and it will call as U tail, like this, U tail. So you have to be really careful about what is the GC hairpin and what is the U tail. So the GC hairpin actually causes the RNA polymerase to actually stop. So just like there is a pin, like press that pin and the process will stop. GC hairpin is like that. So GC hairpin is at the terminator region that will be formed, and it will actually stop the whole process. So if you want to summarize the whole process, and if there is any step that you didn't get, I have already summarized the whole process in front of you. Just take a screenshot and now you are good to go, and we will move on to next step, that is the post-transcriptional modification of messenger RNA molecule. So in prokaryotes, you can say there is no delay between transcription and translation, as the messenger RNA comes from DNA and it goes to the nucleus, right? But on the other hand, in prokaryotes there is no nucleus, right? So in prokaryotes there would be no gap between transcription and translation, but in eukaryotes there is a gap, a bit of gap, there is a delay. Why is that delay? The reason is because the newly synthesized messenger RNA has to be modified into functional messenger RNA, because the transcription actually occurs in nucleus. We are discussing about transcription that is happening in the nucleus, while translation actually happens in the cytoplasm. So the messenger RNA that we just made in transcription has to move from nucleus to cytoplasm. So in eukaryotes there is a delay because we have to move from nucleus to cytoplasm, and during this journey enzymes there are different enzymes like phosphatases, nucleases, they can actually harm your messenger RNA molecule, right? And furthermore, there are eukaryote messenger RNA also have non-coding region. The non-coding region is called as introns. As you can see here, these are introns. 1, 2, these are the introns. What are introns? Introns are actually the non-coding regions. These are the regions that were left behind. There is no code in these regions, so they are kind of like useless. So first thing is that when you have to move from nucleus to cytoplasm, the messenger RNA has to move from nucleus to cytoplasm. The first barrier is enzymes. Like there are many enzymes that are dangerous for RNA, and these enzymes can actually dissolve or destroy messenger RNA. Second thing is introns. They are like the useless parts of like your messenger RNA, so you have to remove these introns too. So to solve these two problems, we have like a process that is called as transcriptional modification, or you can say RNA processes. So RNA processing in eukaryotes actually happens, but not in prokaryotes, because in prokaryotes transcription and translation both happens in the cytoplasm, because prokaryotes don't have any nucleus, right? So there are two events that involve this process. The first process is the addition of a cap. You will have a cap. This is the first solution, and the next thing is that you will have a tail, and then you will also have RNA splicing. So we will discuss about both of these one by one. So first modification is that you will add cap 7-methyl guanosine cap that will be added at the 5' end, that is right in front of you here. So the 7-methyl guanosine cap will be added at the 5' end, and then you have poly A tail that will be added at 3' end. So what does these two actually does? So the cap and tail actually protects the RNA molecule from those dangerous enzymes that were harmful for the messenger RNA. So the cap and tail will actually protect your messenger RNA molecule. And the third modification is actually called as RNA splicing. So what is actually RNA splicing? So you have two types of things here. You have intron and you have exon. So what are exon? Exon actually are the coding regions, and the introns are actually non-coding regions. So exons are the useful part and introns are the useless, or you can say the parts that are inefficient and that are like garbage for you. So what you will do, you will remove the intron parts, you will cut them down, you will splice them, and when you splice them you will have a complete exon like messenger RNA. Only exons will be left and there will be no introns. So you will have a messenger RNA that is completely modified. So this was pre-messenger RNA and you modified it into this kind of structure. Now you have like cap, a tail and a exon molecule. So the RNA molecule is actually now equipped with guns and weapons, right? Before it was like really vulnerable, no anyone could have harmed your RNA, but now your RNA have weapons like guns, like cap and tail, and it has also left behind any useless stuff that it was carrying. Now it is also lighter. So messenger RNA will easily move from your nucleus into cytoplasm, where translation will happen. So this was about introns and exons. This is the difference between them. You can study about them. What are intron non-coding, and exons are coding strand. The introns are actually removed by spliceosomes. Spliceosomes are the enzymes, and exons will remain there, and they are actually really useful. Introns are present in pre-messenger RNA, while exons are always present. Okay, if we about memory trick, introns are like in the trash and exons exit the nucleus. So this was about difference between introns and exons. Let's talk about types of RNA. We have already discussed about types of RNA so many times. I am like sick of this. Like until now we have discussed it so many times. So you can say what is messenger RNA, transfer RNA and ribosomal RNA. So you should be teaching these topics to me now, right? Because we have discussed about types of RNA like I think so this is the third or fourth time that we are discussing these types. But still you have to be really careful about these types and their functions and their names, where are they made and how they are used. The next thing is about the diagram here. So you have to be really careful how actually these molecules actually work. As you can see here, this is actually the process of translation, how translation actually happens by RNA molecules. So you can take a screenshot. We will discuss about it in detail in your translation chapter. The next thing is about the genetic code. Let's discuss about genetics. Like this is the important point you have to keep in mind. So So first is the genetic code. Like you have already discussed in this chapter that gene is a specific segment of the DNA that encodes the synthesis of particular polypeptide protein. Therefore, the order of amino acids, you can say, in a polypeptide is according to the sequence of nucleotide in our DNA. So this is the relationship between amino acid sequence and nucleotide sequence. You can say this is called as genetic code. So the relationship between amino acid sequence and nucleotide sequence is called as genetic code. So you can say the information in DNA for the synthesis of protein is called genetic code in most simpler way. So the information in DNA for the synthesis of proteins is called as the genetic code, which is actually transcribed and copied into the messenger RNA that we just made. So that messenger RNA transcript actually consists of a random sequences of only 4 kind of nitrogenous base. You know what are the nitrogenous bases? You can say these are adenine, guanine, cytosine and uracil, right? So these are the 4 bases. And now the question is, how can a code with 4 letter alphabets can specify a protein like at any given contains like 1 of 20 different amino acid? So only 4 nucleotides are making so many proteins. So clearly like a single base cannot specify a single amino acid, right? If there were a single amino acid specifying a single nucleotide, then it would be only one type of protein from one, and then you will only have 4 proteins in your whole body. But that is not the case, right? So it is also not feasible if only 2 bases are involved. So if only 2 bases were involved, you would have like 4 cross 4 is equal to like 16 possible scenarios or 16 possible codons, right? But 3 bases are sufficient. With 3 bases you will have a total of like 4 cross 4 cross 4 is equal to 64 codons. So this is the possible combinations that you can have from a genetic code: adenine, guanine, cytosine and uracil. So there are specific 64 combinations that are possible by each particular amino acid. So therefore a triplet of base along the length of messenger RNA that specify amino acid is called as codons. What we will call them? We will call them codons. Okay, so you have to be really careful. Like these specific pairings are called as codons. So there are actually how many types of codons? We just discussed it. There are actually 64 codons. Three of these codons are actually called as stop codons that are in front of you. These are the most important ones. I haven't made the whole list of 64 base pairs and their codes because they are not necessary. You only have to memorize these 4 codons. There are actually 64 types of codons, but only these 4 are important ones. You have to memorize these codons. They are really important. And the most important one is actually the methionine. Methionine is actually adenine, uracil and guanine. It is called the start codon, while other 3 are called as stop codons, and they are also really important. So since these 3 codons that do not code any amino acids, you can say they are also called as non-sense codons, right? While all the other sequences that encode specific amino acids are called sense codons. So you can say these stop codons, like 3 of these, they are called as non-sense codons. They are nonsense, right? So they are called as nonsense codons, while the all other codons including methionine, they are called as sense codons. Okay, so methionine and all the other codon sequence, like other 60 codons, are called as sense codons, while the 3 that are in front of you, these 3 stop codons, are called as non-sense codons. So there is really importance of these 4 stop and start codons. Like they have been asked in your IMAT exam about what is methionine, what is stop codon, what is start codon, and this topic is like confusing a bit, but I think if you go through this, you will have a really great value in your IMAT exam. The next thing is about how this whole process actually happened. The first step was initiation, obviously, and then the RNA polymerase actually started binding, and then you had DNA unwinding of the DNA molecule strands, and then RNA synthesis actually began. Then you have elongation, then RNA strand elongation, then termination, and then RNA polymerase actually released, and then you had pre-messenger RNA processing, in which you had added cap and tail, and then you had a messenger RNA that was exported into the cytoplasm. So that was the whole process that is summarized in 10 steps. Even if you like don't get my complex questions, like sometimes I think I might go a bit too deep into the topic, but that is for your better understanding. And if you think like this is too much for me, then believe me, the things that are written in your notes are enough, right? So everything that is written in your notes is more than enough. You don't have to read anything else after these notes. If the secondary source that you have to use is the one that is called as Cambridge or Pearson. Don't read any other books. Don't jump from one book to another book to another book. These are enough. Okay, my notes and my book that you have already seen, the playbooks that you have guys been given, the playbooks contains everything, and the Cambridge or Pearson book. You don't have to use both. You can use either Cambridge or Pearson. Personally, I think Cambridge is contains more theory, while Pearson contains more amount of diagrams. Both of these books are really good. You can use either of them, but the lectures we prepare are actually from both of these books. I prepare from Cambridge and Pearson. I use both of these books as reference when I am preparing your lectures. So you don't have to be like really like thinking about whether we are completing IMAT syllabus or not, what is being left, what if this comes your exam, what if you leave something. Don't worry, we are covering every single thing. You don't have to be worried about anything. Okay, so this is the whole summary for your transcription process here. The unwinding of the DNA, then you had a coding strand and a template strand. From the template strand you made an RNA molecule that was moving from 5' to 3' end, and then this is is called as the terminator region, and RNA molecule is being made and it is being given out from the template strand. So this was like really simple process. So many students make this complex, like we just discussed in your lecture so many things, right? But the process is actually really simple. It's not that complex. You have to keep it as simple as possible, and the simple thing is these 10 steps. These 10 steps are the whole transcription process. You have to keep in mind what is happening in every single step here, and then you are good to go. Okay, so this was all from the transcription process. Then in the next lecture we will discuss about the translation, how translation happens, and then this chapter would be done. Okay, bye bye, meet you in the next topic.